All right, so our last section of notes here has to deal with the final process after we've created the messenger RNA, kind of what happens next. So, and that process is called translation. So, what happens next is that once the mRNA is created, it needs to be read, and therefore then the DNA's instructions will be carried out. This decoding process, like I said, is called translation. This occurs at the ribosome, and what it does is it turns the code into proteins. So we have some pictures here to kind of illustrate this process. So first of all, the messenger RNA strand comes out of the nucleus through these nuclear pores, and it heads out to the ribosome. Now the ribosome actually has two halves. You can see here the blue part and the yellow part. And the tRNA, they kind of look like a letter T. They actually kind of float in, and the ribosome helps them match up. You can see how their anticodons match up with the codons on the RNA, and each of them is carrying an amino acid. So these guys float in, they match up, and what the ribosome does is it actually hooks together these amino acids. And then as the tRNA leaves, it actually unhooks from the messenger RNA, and it goes and finds another amino acid so that it can potentially repeat the process. As this one is leaving, we have another one coming in, and inside, these amino acids are getting joined together. So this process continues, and what we have is a polypeptide, and that polypeptide will get folded and turned into the correct shape. So let's take a look at that mRNA code. Okay, the genetic code on the messenger RNA is actually made up of three-letter words, and it uses the nitrogen base sequence of the DNA, which gets transcribed into messenger RNA. So it creates those codons in the messenger RNA, and each codon codes for a specific amino acid. Now, we have in nature 20 amino acids, and they join together in a specific pattern and a specific number of amino acids to create the proteins, the polypeptides that we have, and we have thousands of them. We have 64 different possible codon combinations. Now, one of the possible codes is AUG, and AUG can code for either the amino acid methionine, or it can act as the start codon for protein synthesis. And we will mark that because it's not marked on one of them, and so we want to make sure that you guys understand that's the start codon. There are 3 possible stop codons as well. So they don't code for an amino acid. So unlike the start codon, which also codes for an amino acid, the stop codons do not. And this is what a typical codon wheel kind of looks like here. And I know you've seen this before. So remember, we work this from the inside out. So we look at the first letter of our codon. So let's, in this case, let's say A. All right, and then we look at the second letter of our codon and we move out to the next level. So in this case, we started with A, we're going to move to U. And then the third codon, or the third letter, we're going to move out to G. And you can see then we land at an amino acid. In this case, it is methionine. I also want you to mark this as start. And you can see we've got different amino acids, and we have the different stop codons that are around. We've got one of them here, UGA, codes for stop, and then the other two are here, UAA and UAG. So some amino acids only have one code. Others have many of them. All right, and we'll be taking a look at this. Now, this is one way that we can look at to translate. This is another way. This is kind of a box way, and it's a little bit more challenging. So what you have to do is you have to start over here with your first nucleotide. Okay, so we're going to start with A. All right, and then you go up here, and it's going to be U, or it could be C, or it can be A, or it could be G. So we're going to say, okay, we're starting with A, and we're going to go to U, and then you can see down here at the bottom we have G, and that codes for methionine or start. All right, if we wanted to do a stop, we could do UAA, or UAG, or UGA. Okay, so we have to start with the first, then figure out the second, and then figure out the row for the third. This one's a little bit more challenging. Mr. Phillips actually calls it the square of dumbness. We like the wheel better because you guys can read it a little bit more easily, but we have seen the square one on the Keystone exam, so it's very important that you do understand how to read this. What it is important to remember is that the protein is determined by the amino acids. So whatever protein we are making is determined by that sequence of amino acids. The order of those amino acids is determined by the mRNA, and the mRNA is determined by the DNA. So it all goes back to the DNA. All right, in your handout packet, we've got one last handout about decoding DNA or mRNA. So we're using the wheel. It says the diagram shows the mRNA codes that correspond to amino acids. Read the diagram from the center outwards. For example, the mRNA code UAC corresponds to the amino acid tyrosine. So find U, and then A, and then C, and it goes to tyrosine. So they filled out the first one. So AAA is lysine. So we've got G, C, G, and that is alanine. GAU is aspartic acid. And the last one is CAA, which is glutamine. Excuse me. All right. So which two mRNA codes correspond to histidine? That is CAC and CAU. How many different mRNA codes correspond to arginine? Now, you have to be careful here. There are 4 down at the bottom: CGG, CGA, CGC, and CGU. But if you go around on the wheel just a little bit to the left, there are 2 more, AAC and AAU. I'm sorry, no, that's asparagine. Arginine is actually a little bit further, AGG and AGA, sorry. So there are actually a total of 6. So not all of the amino acid codons are grouped directly together on the codon wheel. Now this finishes this section of notes. We actually have 2 more sections that we will be doing that are kind of separate topic, one on mutations and one on biotechnology. But the next thing that you will be doing in terms of the sequence is the protein synthesis lab.